Welcome to Lakshman Biology. We are discussing about the topics genetics. Already I have uploaded the two videos which are related to genetics. We are discussing about the terminology of genetics. If you have not seen the previous videos, please watch the videos and you will see later you will see this video. Okay. Next, we are discussing about terminology of genetics. Next, we will see about what is the gene. Gene. Gene is unit of inheritance. Unit. Unit of inheritance. Inheritance. Means that will transfer from one generation to another generation. Genes are transferred from one generation to another generation. That genes are responsible for the transfer of characters from one generation to another generation. Hence, we can say that gene is unit of inheritance. If you observe the DNA, this is the DNA. Okay, approximately 2.2 meters DNA is present in every cell of the human body. Gene is a small stretch of DNA. This is the gene. We can see the gene 1, gene 2, like that many genes we can observe on the chromosome. Gene 2, gene 3, like that. Here, genes are small stretch. Gene is nothing but it is a small stretch of DNA which act as unit of inheritance. The DNA which contain genes combine together, combine with the positively charged proteins and form chromatin. Chromatin later converted into chromosomes. If you see the each chromosome contain the DNA along with the positively charged proteins. Hence, we can say that the each chromosome contain many genes. If you observe the structure of gene, or if you see the parts of gene contain mainly three parts. First one is cistern. Cistern. If you observe the cistern, it is the biggest part of gene. Biggest part and functional unit. It is the functional unit. Functional unit of protein. Gene. Functional unit of gene. It will form protein, which is involved in the translation, and it will form the protein. Hence, we can say that cistern is biggest part of functional, biggest part and functional unit of gene. Cistern we can also call as structural gene. Both same only. Structural gene and cistern both are same only. Second one is recan. If you observe the recan, it is the smallest part, smallest unit. Recan is smallest unit of gene. It is able to recombinate. It is involved in recombination. Recombination is nothing but exchange of the DNA in between the two homologous chromosomes. The recan is smallest part of the gene which is able to involve in the recombination. Hence, you can say that recombination unit. Then mutant. There is another one mutant. Mutant is mutation unit. Mutation. It is able to mutate. Mutation unit is mutant. That is the different parts of the gene. Next, if we see the gene, first Mendel. See, during the Mendel time, Mendel called gene as factor. Mendel called gene as factor. Then if you see the genes, name gene is coined by Johansson. Name gene coined by scientist. Name of the scientist is Johansson. In the year 1909, the Johansson coined the name gene. But the Mendel is belongs to the 18th century. 18th century, he don't know the name gene. He called the unit of inheritance as factor. Only the factor. Next, structure of gene is proposed by Benger. Structure. Structure of gene proposed by Benger. Benger is the scientist who proposed the structure of gene. Already we have seen the structure of gene which contain cistern, recan, mutant. If you observe the prokaryotes and eukaryotes, the structure of gene is different in prokaryotes, different in eukaryotes. What is the difference? Prokaryotic. Prokaryotes means the organisms which not contain nuclear envelope. Nuclear envelope. Nuclear envelope is oxygen. Eukaryotes means which contain nuclear envelope. The prokaryotes example bacteria, archaebacteria, etc. If you observe the prokaryotic and eukaryotic gene, prokaryotic gene contain many cisterns. Cisterns are structural genes. Many structural genes or cisterns are present. Hence, the gene which present in the prokaryotes is called as polycistronic gene. Polycistronic gene. Then, if you observe the eukaryotes, the gene which present in the eukaryotes. Example for eukaryotes: plants, animals, fungus, algae. Okay, except bacteria, cyanobacteria, mycoplasma. Remaining all are the archaebacteria. Remaining all are eukaryotic genes. If you observe the eukaryotes only, that eukaryotic gene contains single cistern, only one cistern. Each cistern can form the one polypeptide chain in the translation process. One polypeptide chain is formed from each cistern. If you see the prokaryotes which contain many cisterns, hence can form more polypeptide chains. The gene which present in the eukaryotes is called as monocistronic gene. Monocistronic gene. Monocistronic. The gene which present in the prokaryotes is called as polycistronic gene. Next, we will see about alleles. We have seen about the gene. Next, we will see about alleles. If you see the gene, mostly each gene contains two alleles. We can say that it is a pair of contrasting traits. Alleles, alleles are nothing but pairs, pair of contrasting traits. Pair of contrasting traits are called as alleles. We can also say that forms of a gene. Alleles are forms of a gene. Mostly, if you see the each gene contains two alleles. That is both dominant or both recessive, or one is recessive, one is dominant. Okay, gene contain two alleles, dominant allele and recessive allele. Dominant and recessive allele. That is about allele. Example, if you see there is a gene which contain two alleles, dominant allele is capital A, recessive allele is small a. Next, what is the dominant allele? Dominant allele. The allele which express the phenotypic character. See, dominant allele means allele that express phenotypic character. Even in the presence of an alternate allele. Here, dominant allele means allele that express phenotypic character even in the presence of an alternative allele. Example, see there is an organism which contains two alleles. One is dominant allele, one is recessive allele. This is the dominant allele. Then there is another organism which contains two alleles, both are dominant. There is one organism which contains two recessive alleles. Here, if you observe the gene which I have given as example will maintain the height. Phenotype means phenotypic character. Phenotypic character means the character which will appear externally. That is because of gene which present inside the organism. Here, there is a gene which is responsible for the maintenance of height of the organism. If you see the first one, in case if it is present in the tall, we can see the formation of tall plant. Here, dominant allele is responsible for the tallness of tree. This dominant allele will express its phenotypic character. Phenotypic character here is the tallness of plant. even in the presence of an alternative allele. Alternative allele is small t. Alternative allele is small t. Capital T, small t. Okay, will give the tall plant. Then s- capital T also give the tall plant because both are dominant. When we can see the dwarf plant, when there is both recessive alleles. Recessive allele here express its phenotypic character only in the homozygous condition, only in the presence of another same type of allele. But if you see the dominant allele, which will express its phenotypic character even in the presence of an alternate allele. Next to recessive allele. Recessive allele. See, recessive allele means express phenotype only in the presence of an, another identical allele. In the presence of another identical allele. The recessive allele expresses its phenotype only in the presence of another identical allele. Example, if you see small t, then only you can see the formation of dwarf plant. Dwarf plant. If there is a gene which contains the two alleles, that is small t, when both are similar alleles, then only you can see the expression of the phenotypic character. Example for that is small t will give dwarf plant. Dwarf plant. That is about recessive allele. Recessive allele. Remaining we'll see in next class. Thank you.